Hello everyone, welcome to Biology with Medmelo, and today we are going to continue the question of our DNA. That is, we are going to learn the concept of DNA replication. So by literal meaning, the definition of the DNA replication is the process of replication of DNA into two new daughter DNA which are identical to one another by using certain enzymes. The method of DNA replication is a semi-conservative method which was postulated by Francis and Crick and scientifically proved by Michelson and Stahl. First of all, please put some stress over there that postulated and proved by are different. Postulated, that is, in 1953 when Francis and Crick proposed a double helix model of DNA, they also suggested that during their observation, they noted that DNA can replicate in a semi-conservative method, which was later on proved in 1958 by experiment of Michelson and Stahl, in which they used two isotopes of nitrogen, N14 and N15, on Escherichia coli. So we can say that the semi-conservation method was basically proved, and foremost credit goes to Michelson and Stahl. Now, what is the semi-conservation method? Semi means half and conservation means sustained or conserved, which means if there is a DNA like this, and if it is going to replicate, first of all, it will divide itself into two, right? So these are your parental genes. Now the new DNA strands will be going to attach to themselves and they will form a new DNA. So in this case, what we are seeing that these kind of genes are what? The parental genes, which are conserved in halfway. That is, they are semi-conserved in their new daughter cells. Okay. Now this DNA replication involves 4 enzymes. I have given them 4 names according to their function. First of all, we have the unzipping enzyme, which is DNA helicase, that unzips or open the DNA fragment at the replication site. Then we have the builder enzyme, that is DNA polymerase, which is going to make the new DNA daughter strand. Then we have the primer enzyme, that is the RNA primase, which will show the way to the DNA polymerase. That is, the DNA polymerase will only act when it will be notified by the RNA primase that where you have to start. Then we have in the end the gluing enzyme, that is the ligase enzyme, which will bind everything all together. So we have 4 enzymes: the unzipping enzyme, DNA helicase, the builder enzyme, DNA polymerase, the primer enzyme, RNA primase, and gluing enzyme, the ligase enzyme. Now the process of DNA replication is divided to 2 stages. First of all, we have the unwinding at the origin of replication. And second, we have the formation of new strand. Now let's discuss these processes in this DNA. First of all, we said that there is unwinding at the origin of replication. Now what is this origin of replication? In the previous video, I told you that the hydrogen bond between adenine and thymine is weaker than that of cytosine and guanine. Suppose here is a bond formation between adenine and thymine. So this bond is easier to break by the DNA helicase enzyme. So he will start working over here to open the DNA sequence. This sequence is known as origin of replication because it is foremost the easiest part to part the DNA strands. Okay, now the DNA helicase works is over. Now we call upon the DNA polymerase. DNA polymerase is a very conditional person who works upon 2 conditions. First of all, the DNA polymerase, who is basically responsible for making the new strand, will only form the daughter of polarity 5' to 3', which means we must have the parental DNA of the polarity 3' to 5'. That is, he will start working from this strand. Now, how will he get to know that where to start or which strand is 3' to 5'? Here we get the RNA primase. RNA primase will make a signal over here that you have to start from here, and then the DNA polymerase will start working over here by making a daughter strand of polarity 5' to 3'. Now, when this daughter cell is making, or the daughter DNA is making, two strands are made by the DNA polymerase. The one is the leading strand, the other is the lagging strand. How? You can see that this strand is continuously duplicating without any fragments. This strand is known as the leading strand, while this strand over here is basically have short fragments which have spaces in between them. This strand is known as the lagging strand, and it is much weaker than that of the leading strand, so it may need one another enzyme to make it up a new DNA strand or glue it up. Now these fragments are made up of 150 to 200 base pairs, and these are known as Okazaki fragments. Now we need the enzyme which can glue up these empty spaces. Hence, we here need what? The ligase enzyme, who will fill up these spaces with what? With phosphodiester bond. He will glue these fragments with phosphodiester bond so that they make a complete new DNA daughter strand. So here a complete new daughter cells are made with one strand of their parental gene. That is, they are hence proving the semi-conservative method of DNA replication. So let us summarize what happened in this shell. Okay. First of all, the unwinding and unzipping of the spiral DNA by DNA helicase enzyme at the origin of replication of adenine and thymine bond is broken. The unzipped sequence perform the matrix roles and perform the Y-shaped fork, which is known as the replication fork. Now the daughter sequence of 5' to 3' polarity is made along the mother sequence of 3' to 5' polarity by DNA polymerase. And DNA polymerase will form two strands, leading strand and one lagging strand. The leading strand is continuously duplicating in the direction of 5' to 3', while the retarding strand is duplicating itself in short fragments, which are known as Okazaki fragments. And these are running opposite to the polarity of the leading strand, that is 3' to 5'. The linking of Okazaki fragments in the end is done by ligase enzyme through the help of phosphodiester bond. So this was all about the question. I hope you understood it well, and if you have any doubts, please let me know in the comment section. Thank you.